Hello everyone, I hope all you are fine. Today we are in chapter 10, which it is molecular genetics. In this video I will try to explain the questions which are related with this chapter, which it is molecular genetics. Molecular genetics, it means simply studying genetics at a molecular level, such as studying the nucleotides of the DNA, amino acids of the proteins, okay, and the mutations. But in classical genetics, we are depending on the traits, phenotypes, colors. But in molecular genetics, it is more accurate and it's more convinced. So now let us start by solving the question that we have in this chapter. So the first question that we have, it says all of the following about the structure of DNA are correct except. So we have 5 choices from A to E. Okay, 4 of them are true, only one of them that's not true. It's not related with the DNA, okay, with the structure of the DNA. The first one, it says DNA is a polymer. Yes, DNA is a polymer and it's made up of many monomer units, and these monomer units are nucleotides. So the first one is true. DNA contains deoxyribose. Exactly, when we say DNA, okay, this D, it means deoxyribose sugar, while in RNA we have ribose sugar. Okay, so DNA contains deoxyribose and RNA contains ribose sugar. Okay, so this one is also true. The two strands are connected by hydrogen bond. As we know that we have two strands of DNA. Okay, these strands are connected by hydrogen bond. Okay, the two strands are connected by hydrogen bond, while the nucleotides of the same strand, okay, of the same strand, okay, not with this strand, this is the hydrogen bond, okay, but the nucleotides of the same strands, for example this A with this thymine, okay, they are connected by phosphodiester bond. Okay, it's not hydrogen bond. So the C is also true. Adenine bonds with guanine, I think it's clear that in which this one is not true, as we know that adenine makes pair, or we can say makes a bond with thymine. Okay, so this is not true. Let us read the last one. Nucleotides consist of sugar, yes, phosphate and nitrogenous base. Exactly, each nucleotide that you have is made up of a sugar molecule, a phosphate and a nitrogenous base, okay, in which it may be either adenine, thymine, guanine, or cytosine. So the answer for this question is D. Okay, so all the others are true, okay, about the structure of DNA except D. So the answer for the question is D. Okay, now let us come to the second question. Okay, in the second question, it says if guanine makes up 28% of nucleotides in a sample of DNA from an organism, so we have guanine, okay, it makes up 28 percentage, okay, of the nucleotide in a sample of DNA. Then thymine would make up how much, okay, percent of the nucleotide. So we have to solve this question. As we know that guanine makes pair with cytosine. So if guanine is 28, then cytosine is 28 too. Okay, so 28 plus 28 equal to 56 percentage. Okay, so the amount of the guanine and cytosine is 56, okay, of a DNA sample. So now we have to come to that. So the remaining part, okay, 100 minus 56, it will be 44. Okay, so 44 for what? 44 for adenine and thymine. Okay, because we know that adenine and thymine are pairing with each other. Okay, so 44 for all of the adenine and the thymine in that sample. Okay, so you will divide it by 2, it will be 22 percentage of adenine and 22 percentage of thymine. So the answer for the question, as we can see, and it's very clear, it is B. Okay, so I hope it's clear. And now let us move to the third question. It says all of the following statements about RNA are correct except, yes, let us see. There are three kinds of RNA. Yes, we have messenger RNA, ribosomal RNA, and transfer RNA. Okay, so this is true. RNA stands for ribonucleic acid. Exactly. RNA, it means ribonucleic acid. Is a single strand. Yes, it's a single strand, while DNA is a double strand. RNA contain the nitrogen bases cytosine, guanine, adenine, and thymine. But we don't have, pay attention please, we don't have thymine inside the RNA. Instead of thymine, we have uracil, okay, that pairs with adenine, okay, during the translation process it will be substituted with that one. Okay, so we don't have thymine. Okay, so this is the right answer for the question 3. Okay, but let us read the last one. It contains sugar ribose. Yes, it contains sugar ribose. So all of the following of the statement are correct except this one. Okay, this is the answer for the question. So in the fourth question it says DNA can be cut into small pieces with what? How we can cut the DNA into small pieces? So guys, very simply, we have restriction enzymes. Restriction enzymes, they are like scissors, okay, in which they are cutting the DNA at a specific nucleotide. Okay, so by the restriction enzyme we can cut the DNA into very small pieces. Okay, so the answer for the question is D. But let us know what are the other terms that we have. We have exons and introns. Okay, as you know that exons, they are the coding part of the DNA. Okay, they are the coding part of the DNA. Okay, while the intron, they are non-coding part. PCR, it's polymerase chain reaction. It's a reaction in which it is done by an instrument we call it thermal cycler. Okay, in which in the lab we can amplify or copy the DNA that we have, and you know that what's RNA. Okay, so just know that restriction enzymes are those enzymes that they cut the DNA into small pieces. So in the fifth question, it says if the sequence of a strand of DNA is AAATAA and so on, then the sequence of the corresponding mRNA. So pay attention. It is mRNA. Okay. So inside the RNA, we said that we don't have thymine. Okay. So instead of thymine, we should have uracil. Okay, so before coming to these choices, okay, now let's just do it by ourselves, guys. As you see, inside the mRNA, for this adenine it will be uracil, 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 and thymine will be adenine, uracil, uracil, guanine, guanine, cytosine, 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 cytosine. This A, okay, it's in an RNA, it will be uracil, thymine will be adenine, cytosine will be guanine. Now let us see which one is true. So the answer is UUUAUU. Okay, so which one is like this? As I see, this choice, okay, it's UUUAUU. It is the same that I did here. Okay, so this is the answer for the question. Just you have to know that inside RNA we don't have thymine. We have, instead of thymine, we will have uracil. So I hope it's clear, and it's very important. You have to know, and it's very easy. So in the sixth question, it says all of the followings are correct about RNA processing, except it says it's a rare occurrence and results in mutation. Okay, introns are removed. Yes, we know that during RNA processing, introns, usually they are non-coding parts. Okay, they will be removed because they are not required, but occurs after transcription. Exactly, when the transcription is finished, then the RNA processing will start. It's occurring after transcription. The initial transcript is longer than the final transcript. Exactly, if the first or the initial transcript is like this, okay, then the final transcript, because of the removing of the introns here, okay, so it will be shorter. Okay, it occurs inside the nucleus. Yes, it occurs inside the nucleus. So as you can see, the answer for the question will be A. Okay, A is the answer for the question, because it says it's a rare occurrence. It's not a rare occurrence. After each transcription we have this process, and it's not resulting in mutation. It's not related with the mutation. Okay, so I hope it's clear till this point, and I think it's enough for this video. In the second video we will continue from the question 7, and thank you so much for your listening.